Okay. Can you see my screen?
Yes.
Okay. Um, Thank you. Good evening. I'm Jessica Lucchesi, a PhD student at Lens in the University of Florence. And today I will talk about my PhD project entitled In Vivo Calcium Imaging of Mice Engaged in Social Behavior Reveals Widespread Interbrain Synchrony. Okay. Social interaction. Social interaction is a term that represents the natural disposition to interact or associated with one another. And behavior can be influenced by another individual or by environmental factor. And we could say that social behavior arises as a result of an interaction between two or more individuals and the surrounding environment. And the mechanisms involved in social behavior are extremely complex, but are important for the health and the well-being of every individual. Humans, by definition, are a social animal, but when social interactions are compromised, are a clear sign in several psychiatric disorders such as autism, schizophrenia, depression, and the social anxiety disorder. To study social interaction and the mechanism underlying behavior, a good animal model to start are rodent. Mus musculus is a social species that exhibit reciprocal social interaction, sexual and parenting behavior, territory marking, aggressive behavior, and in addition tend to lead in groups by setting up hierarchies. And given their wide behavioral repertoire, rodents are the appropriate species to study social interaction under both physiological and pathological conditions. About the neural mechanism, the mechanism that coordinate behavior and social interaction are largely unknown. But in rodent, the cortical areas most involved in modulation and control of social behavior is the medial prefrontal cortex. From literature, research show that a subset of neurons in the medial prefrontal cortex exhibit elevated discharge rate while mice approach an unfamiliar mouse, and this doesn't appear when mice approach an animate object or, for example, an empty chamber. And all of these observations are consistent with the idea that neural activity in the medial prefrontal cortex correlate with the social behavior in mice. But in addition, in mice brain, are there another structure that are involved in social interaction? For example, about the subcortical structure, we can find amygdala that is implicated in emotional processing, hypothalamus that with the production of different hormones modulate the stress and the behavior of the animal. And the most important hormone that modulates social interaction is oxytocin. We can find hippocampus for the memory processing, and in the last year we emerge nuclear accumbens for social incentive, kindness, and another social behavior. And last but not least, the importance of olfactory system, which play a key role in the mechanism of interaction and exploration in rodents. In the past, several paradigms have been described to measure behavior, to measure social behavior in mouse model. And among them, the most uncommon is the sociability test, also called the three-chamber test, developed by Crowley's group. And in this behavioral paradigm, after a first phase of habituation with one mouse and the other side of the chamber, the empty chamber, we test the animal preference between an empty chamber and an unfamiliar mouse. And finally, we test the animals for the social novelty with the introduction of a second stranger mouse. But what are the techniques for studying neural activity? Techniques most used to study neural activity in rodent are based on electrophysiology recording, and these allow us to recording single neuron activity through the implantation of one or more electrode in brain areas of interest. However, this technique has the disadvantage of recording a very small brain region. So we have the information about the neurons that we can find near the electrodes. And in the last year, optical calcium imaging has been remarkable development. In detail, thanks to the use of a transgenic mouse model that expressing calcium-sensitive fluorescence protein, we can record, for example, using a wide field microscopy, neural activity across large area of the cerebral cortex and for a long time. However, these microscopy techniques require head fixation, and a long time for acclimatization is necessary, and this head fix condition limiting the animal's behavior repertoire. So what is the new era of optical imaging? New era is based on Miniscope. Miniscope is a miniaturized head-mounted imaging device that are able to monitor neural activity in freely moving condition. Here I will show some different type of miniscope developed by various universities of USA. Miniscope present different feature and design, but as we can see, most all of these present a small field of view, so allow studying neural activity at a cellular level with over a small brain region, and also to have the access of this small brain region. In most of the cases, a craniotomy is necessary. So all of these precludes the study of neural dynamic distributed over large cortical area associated with complex behavior like social interaction. So after this background, we know the importance of social interaction for the health and the well-being of every individual. And we know the behavioral test to study social interaction and the importance, the role of the medial prefrontal cortex in rodent and in humans, and the subcortical structure. And at now we have some device, some miniscope to study neural activity at cellular level, but in freely moving condition. So after this background, the goal of our project is to develop an innovative optical method for imaging neural activity on both hemispheres in freely moving mice engaged in social interaction. And also we validate a behavioral paradigm to study this phenomena, looking for a possible neural correlate of social interaction. So in this slide, I will present our miniscope, the design of our miniscope that is mounted on the head of transgenic mice that express GCAM6F indicator in a subset of excitatory cortical neurons to monitor, and this device is able to monitor bilateral cortical activity through the intact skull, so a craniotomy is not necessary in this case. The body of the miniscope was built using our stereolithography 3D printer and was equipped by two blue LED with 470 nanometers of wavelength to excite GCAMP indicator paired to excitation filter. And then the fluorescent signal was filtered by emission filter and recorded by CMOS spy camera. In our study, we investigate the social interaction between mice in a custom-made environment. In detail, this arena is composed of two chambers separated by a transparent wall with a fissure located in the center to allow the mice to interact with the nose. The experimental paradigm has foreseen an initial phase, an habituation phase, in which the single subject with the miniscope on its head was placed alone into the chamber for 5 minutes to explore the environment. And finally, in the last session, both subjects were placed in their respective chamber to record the social interaction session. and behavior and cortical activity during all the experiment were monitored using a Raspberry Pi system. To validate and quantify social interaction, we performed ROI-based analysis using a custom-made algorithm to determine the number and the duration of no-spoken interaction when the subjects are alone, indicating in orange, and when both subjects are present in yellow. And the duration and the number and the duration of no-spoke are greater when there is a second subject on the other side of the arena. So might spend more time near the gap when a second subject is present. And also, on average, the first no-spoken interaction is longer compared to the subsequent. To study cortical activity in mice engaged in social interaction, we segmented the behavior and we divide the behavior into different groups. First of all, I will present the interacting dyad. And for the interacting dyad, we divided in gap interacting and gap solitary. Gap interacting is a condition when both subjects are present and goes simultaneously to the gap to interact with the nose. While gap solitary is a condition when both subjects are present, but only one goes to the gap and the other companion doesn't interact. And the second behavioral categories is surrogate dyad. Since in our experimental paradigm, the social session was preceded by 2 preliminary sessions where the subjects are alone, we decided to use this recording to generate a surrogate dyad as a behavioral control. In this recording, the behavior of the single subject was segmented, selecting the moment in which the subject goes to the gap and the other side of the chamber is empty. And subsequently, the session of the individual subject were artificially combined to simulate the interacting dyads. Thanks to the miniscope, we are able to record the calcium signal profile expressed as delta F over F from both hemispheres, and we used it to measure the coupling between the interacting dyad. And to study a possible coupling of these 2 signals, we used the wavelet coherence transform, which allow us to measure the synchrony or the level of coherence between 2 time series as a function of frequency and time. So once the wavelet coherence transform analysis has been performed, we obtain a spectrum in which we find the duration of the recording and the different frequency band related to calcium signal profile, and in colorimetric scale, the level of coherence. And from the spectra, we cropped the time window in which the interaction for gap interacting and gap solitary took place. So we cropped this time window and then we extract the full spectrum of coherence. And the same thing was done for the surrogate dyad, a condition in which the subject goes to the gap, but the other chamber is empty. And after this, after this segmentation, we obtain the full spectrum coherence distributed over the entire cortex. These data are obtained by averaging 11 interacting female dyads. And in the graph, I will report the level of coherence in different frequency band in 2 behavioral conditions, interacting dyad compared to the surrogate dyad. And we can see that only in the interacting dyad, we observe an increase of coherence in 2 frequency band, one at a low frequency and the second at a high frequency. And this phenomena doesn't appear during the surrogate dyad. So after this, we decide to analyze another behavioral condition. So the solitary dyad compared to the surrogate dyad. And in this case, we observe an increase of coherence at a low frequency band, but we doesn't observe an increase of coherence at high frequency for the solitary dyad. And also we observe that the level of coherence at low frequency is lower compared to the interacting dyad. So the presence of the subject on the other side of the chamber is sufficient to determine an increase of coherence. Here I will report the coherence variation, defined as the difference between the coherence during gap social minus gap surrogate, in the first instance for the whole cortex and then for parcel brain region. And after this analysis, we observe the increase of coherence at low frequency and high frequency remain. But when we analyzed the coherence variation into different brain regions, so in our study, I will decide to divide the whole cortex into 4 macro areas: motor, somatosensory, associative, and visual area. And in this case, the high and low frequency coherence peak remain only in the somatosensory and visual area. And this is more visible in the next slides when we analyze the coherence map during this behavioral condition, gap interacting and gap surrogate, only in these 2 frequency band. And we can see that the increase in the increase of coherence at high frequency band is present in visual and somatosensory area. So in conclusion, during this experiment and this first preliminary analysis, we observe an increase in high synchrony during social behavior, and this interbrain coherence is modulated by the type of social behavior at the gap. So we have the measure coherence during the interacting dyad and compared to the solitary dyad, and we doesn't observe an increase of coherence during the surrogate dyad. And also when we analyze the different brain regions, we observe an existence of map of coherence. So maybe not all of the regions contribute in the same way to the coherence. In conclusion, I would like to thank my mentor, Alessandro Scaglione, for his great support and collaboration in this work, and my supervisor, Anna Letizia Allegra Mascaro, and Professor Francesco Pavone. Without all of them, none of this would have been possible. And of course, I thank all of you for your kind attention. Thank you.
Thank you, Jessica.
Thank you.
There are questions? Have questions? I have one question. Which kind of experimental apparatus you used to make calcium imaging? You used the 2-photon technique?
No.
Or classical Fura method?
Now, thanks to Miniscope, we conduce a wide field imaging, so is 1-photon microscopy techniques. And we record calcium activity from excitatory cortical neurons that express these GCAM6F indicators. So when the level of calcium increase into the cell, with calcium binding these fluorescence protein that emit fluorescence, and so we recording the fluorescence signal using this miniaturized 1-photon microscopy device.
Okay, thank you.
Jessica, can I make a question?
One moment, there is one question here.
Okay.
Okay, yes.
Just a curiosity. The sex of the mice, the age, and if they've been alone or in a group before the experiment.
Okay, we chose female mice, female subject, to increase the possible interaction and to avoid aggressive behavior. And in general, to conduce social interaction experiment, the best condition is to use a younger mice. So the maximum range of age is 6 months. And we, in our study, we use animals that are not sister. So we test animals that are not parenting and are stabulated alone. And we isolate the animal in different wardrobe after the social interaction to avoid olfactory stimulus.
Okay. Paolo, please.
Yeah, so no, I have a question about the increasing coherence that you find when they are interacting. Do you have a theoretical interpretation about that increasing coherence?
Okay. We think that the increase of low frequency band is also being seen in human during cooperative and competitive tasks. So we think that the increase during low frequency is modulated by the interaction. While the increase during a high frequency band, we think that is correlate with the communication between the different brain regions. So it could be linked to how the different brain areas are coordinated.